Howdy.
Huh? What?
Okay. Ockert Greyvenstein joined Ball Horticultural Company in 2014 as plant breeder with PanAmerican Seed. A transplant from South America, he came to the U.S. for graduate school, first earning his master's at Cornell, following by a Ph.D. in plant breeding from Texas A&M University in 2013. Currently, he is the regional breeding director based in Elburn, Illinois, overseeing breeding operations in Illinois and Costa Rica with projects ranging from Angelonia to zebra striped tomatoes. During his time with Ball, he bred several crops including Viola, Vinca, Cuphea, cut flower, Delphinium, and a few others. His expertise is in breeding and applied research. Outside of work, he enjoys spending time with his wife, who he met at Texas A&M, two children, and a yellow Labrador retriever. Aside from getting their garden up to standard, his current priority is getting both his fourth and second grade son and daughter self-sustained with fishing, such that he can one day fish again. He would probably also have been happy with a career in sheep breeding or as a mushroom forager and loves reading and dabbling in those fields. Oakert enjoys opportunities to support students and their journey, learning from them and their project feeds his curiosity. Plants remain as fascinating to him as they were when he first tried to graft onions and agaphantis as a 5-year-old. Everybody.
Hey, thank you, Ivan. Coming this way around. I guess I get started.
You're late.
Can everybody hear me okay if I speak like this? Okay, good. So like Ivan said, I'm a regional breeding director out in Illinois working for Ball Horticulture, and I graduated in 2013. And just one quick correction, South Africa is my origin. So I'm gonna get right into it, and maybe before I get topic or my main heading is making luck stick, and I'm hoping by the end of the talk that you'll understand how I got to this point. I also want to say just it's easier for me if we treat this as a conversation and not just a I don't wish to just speak at you the whole time, so let's talk together. So if you have questions, please feel free to ask right away. This is the email I got from Mason as far as the instructions. So it's the series here is where are they now, and so I'm going to take you on that journey, and hopefully you guys find value out of that. It's a little ambiguous, the ask. So the ask was quite ambiguous. So
Hey, we talked after that.
we spoke, but I still think that so what I thought I'd do in case I lose you and you fall asleep during the talk is I'm gonna start with the conclusion that I've reached so far in my career, and the main points that I'm hoping to share here is that for what I've seen so far in my career is got to stay curious. Maintaining a healthy curiosity, probably why you were in university in this department, and if you're going to graduate school, you're probably curious by nature. So maintain that curiosity. The other one is that luck. We all will experience good luck, bad luck throughout our careers, and those events can have outsized impact on your trajectory. The other thing is the little ant I put on there as an icon. I had a beaver there, which I thought was a squirrel, and then my wife was like, just use something different. And so what I'm trying to say with that, that's a long-winded way of really saying that just work hard. So do things that put you in position to make good luck stick, but you do that by getting the job done and working. The other one here that I want to talk about is the people icon. And this part took me an embarrassing long time to fully realize how important it was. I was very focused on the science before. And of course, doing good science is what got me in the door. But what will help you grow is the more important part is people. So if I was here maybe 2 years after graduating, I think the number one thing I would say that was missing from my education would have been something on the business side. So I understand the business aspects, I think it's important to know what you're doing and how you're doing fits into the business model in the organization that you're at, even at the university, that counts for it. Well, where I'm now, it's very clear that that the people part. So if you get an opportunity to, it doesn't come naturally to you, or even if it does, finding ways to understand how we interact, because we're complex beings, and I still fail at this on a daily basis, but that's important. And then the last one here is, I like to have goals, and I like to go for them and write them down and do all of those things, but I've also learned that every
Something, things change.
exactly every single one of those will probably change, and your timelines are going to change. And throughout the presentation, I'm going to put out some highlights of achievements and events. The intention is not to impress anybody in the room with those. The intention of those is to support my conclusions when I do that. So this is just going to be the outline of presentation. We'll start with early beginnings. So where did I start? Education, and then in professional career. I see myself as here in full flower now, so I still have time to get to seed set and harvest. But so that leaves room for changing my conclusions later, but this is where I'm now. And when I talk about my professional career, I'm going to just zoom out a little bit from myself. We'll focus at breeding at Ball just in general, my time at Ball. And after talking to Professor Dingra, he said maybe throw in a little bit something of the science. And so I'm going to talk one specific aspect of one of my breeding programs, which is orange, something with orange violas. So let's just jump right into it. So growing up, I was born in the Highveld of South Africa and spent most of my life on a small farm close to the administrative capital. Talking about luck, any kid's dream was to grow up on a piece of property surrounded by plants and animals. The rule in the house was, if the sun is shining, you're not inside. And so I rarely got to watch TV because there's about 2 days a year that the sun doesn't shine where I grew up. Never too hot, never too cold. This is a 11th grade photo there. I couldn't find something at 12th grade, but pretty much at this point, I was dreaming about being a sportsman, a sheep farmer, and a genetic engineer. Those were basically my dreams. I didn't know how to make them work. I did have to throw this photo on here, and so certain people have big impacts on your life. My grandma was a florist at some point, and we always had a big flower garden around, sweet peas. This was them at their 50th wedding anniversary. And my granddad, we had a big veggie garden on about probably half an acre or more even throughout the time when I was little. And so I still do today love growing potatoes and pumpkins and things like that. And that's just something that, they added to my journey. And I'm throwing the sheep in here because they actually play a big part. This ram was actually one I bought a year and a half ago, and my mom just sold it yesterday without previously talking to me. I was very proud of owning him. But when I was about 10th grade, one of our neighbors dropped off 4 animals, 3 ewes and a ram, and I just fell in love with these native sheep. And the idea of breeding wasn't novel to me. My dad is a geneticist. He ran a dog breeding operation, and he was a corn and sorghum breeder previously in his career. So I had exposure to the idea of breeding growing up. And so what I thought, and if you look back at this animal here, if I wanted to make a little bit of money out of these animals, I should sell genetics. And the top price for genetics was selling animals with multiple horns. So basically breeding ornamental sheep. That was that was the goal. And that helped me from never needing a service job going through undergrad. I coached sports and sold sheep basically for my beer money. But so quickly, just my basic education. I went to the University of Pretoria, got a bachelor's in biotechnology, honors in genetics, worked in the population genetics lab there. And all through my summers in undergrad, I worked with sunflower breeders, oilseed industry. And that's where I got a real taste for applied breeding. And at the time, working on fig wasps, I graduated in 2005. This was published in 2013. So I'm just talking about timelines not working out the way you think it should or want it to. And the reason I put this photo playing cricket, I went to Ireland for a season to play and coach. And why this is important is because growing up where I did, small town, little place, I would have never considered international opportunities without having gone abroad to do something else. At this point, coming back from Ireland after taking a break, I was going to become a veterinarian. I had one credit hour, one class to do in animal science, so I could move into vet school, but I still give my mom a hard time for that. And so I landed on a Sunday night. The plane landed about 8 at night. So it was around somewhere between 9 and 10 in the evening, driving back to our house. My mom explained to me that you have an interview tomorrow morning. And her reasoning was that, well, you're a grown up, you're not a student, you don't have a job, and if you want to live at home, you're going to pay rent. So you have a job interview tomorrow. She knew me better than apparently I knew myself, because she said, you're good with plants, you know stuff about plants. So I got a job as a grower with this wholesale nursery. And that's where I first started seeing about the magic that came out of these green foil package. And that was my first introduction to balls, so a small little ball there. I was just fascinated with the magic of what comes out of these packets. So the veterinarian dream started slowly moving away, but I just did a search to do flower breeding, and 2 universities popped up. The first was Wageningen, and the second one was Cornell. I emailed both. Till today, I've never heard back from Wageningen, but Mark Bridgen at Cornell reached out back to me, and I owe a great debt of gratitude to Mark for taking a chance of me, being quite creative in how he was able to fund me and get me to the US. I probably had the lowest GRE score possible for Cornell to admit anybody. But he took a chance on me. I was happy about that. My research work there was in Plectranthus and tissue culture. But I threw this up here because it's funny. If you look on Google Scholar, the first thing that shows up under my master's work is actually the little snippet about sheep, which I find that amusing. And again, I have to bring it up because I asked Mark, why did he talk to me, or why did he respond to me? And said at that point, he was receiving emails regularly from international students wanting to come. And somehow his eye caught something about breeding ornamental sheep. And that was the hook that made him open up and read the rest of what I wrote. And so, again, sometimes you don't know when luck strikes, but if you're curious and you work at it and you are genuine with the people part of it, things change unexpectedly. I was always quite disappointed that I never got proper publications out of my master's work, but what did happen, I lived above the tissue culture lab at the Long Island Research Station for 2 summers, and my work was quite tissue culture heavy. Mark is actually known as a Alstroemeria breeder, and I just went out in the mornings and made Alstroemeria crosses, and he said, that's great, do what you want there, as long as you do the embryo rescue and the initial tissue culture to get these plants established. And I said, no problem. And lo and behold, in 2025 in January, we got a plant patent out of some of the work we did in somewhere between 2007 and 2009. And it was at this time also where I met somebody from Ball for the first time at a Sealy conference. I met Dr. Charlie Hall there, and I met Marvin Miller from Ball. And so now I knew somebody at Ball. Because I still remember the seed packets. And then I really wanted to stay. This is a busy slide. We don't have to read everything. I'll highlight and apologize for it. I see now how busy it is. But I really wanted to stay at Cornell. I couldn't secure funding, and my advisor, or one of my committee members, said, well, one of my former graduate students is down at Texas A&M, and he may have something. And that's how I got connected with Dr. Byrne and Dr. Terry Starman, and we're able to secure funding through the Monsanto Fellowship in Plant Breeding. And that's how this journey started, working in high temperature susceptibility in garden roses. And the main goal was to try to understand how to phenotype high temperature stress in roses, because if you can measure something, you can breed for it. Otherwise, you're unlikely to make any breeding progress. And Oscar and Ivan, you guys are still working on some of those things. This was probably not the best work that's ever came out of this department, but what it did allow between Dr. Starmer and Dr. Byrne, they allowed me to do my own research. Obviously, a lot of guidance came with it, but there's a lot of learnings that comes from that. And it's so important to publish your work. I just threw 2 publications out here. When this first one got accepted, this is the first time when I actually didn't feel like an imposter anymore. I felt like a real scientist when something in a proper journal was accepted. And this one down here, I was actually quite proud of. And Dr. David Riley is here from the genetics and animal breeding department. Some of what was published in here was because I took a class with him and we learned about power analyses. And I thought that, and this paper ended up getting a Kenneth Post Award that year. And I believe it was because the power analysis that was added into the work there. So point of that is do some cross-pollination. Don't just stay within your department. Go learn something somewhere else. And then again, these silly sheep show up again in my life. There's a constant part to it. I reached out to Dr. Riley before coming to A&M to ask if he would allow me to take some of his classes in animal breeding, which he graciously allowed me to do. And a couple of good things came out of that. I met my wife my first week at A&M. She, well, at that point, a girl I really liked, but later became my wife. She was one of Dr. Riley's students, and then met Dr. Riley. And at some point, they brought Dr. Ben Hayes over, world-renowned in his field in genomics. And my wife was his chaperone to drive him to and from places. And I happened to bum a ride on the way to Lake Bryan to get a couple of free beers and a nice lunch out of the whole deal. But here I had a captive audience with Ben Hayes. Turns out he finds sheep interesting. He thought it was cool that there were sheep with all these horns. And by the end of the day, we had what we thought it might look like to do a small project together. And why this was important, if you're doing your own research and you need to publish that as a grad student, you're basically doing everything. Everybody here knows you. You're doing the experiments, the analysis, the literature studies, the writing, which you have to do to learn. But what this allowed me to do was a truly collaborative project. One part of the team did the analysis, somebody else did the phenotyping and DNA extraction, and we all wrote together. And what this helped me to do was understand better how to work collaboratively across time zones and cultures and still come up with a good result. So that's why I'm putting that out. And then this little bowl sign here, I'm going to talk about luck again twice. I met the head of HR for Ball here when he was recruiting for undergraduate students. In my second year, in my third year, he was down here again, just stayed in contact. And when it was ready to graduate, I called him up and asked if he had a job. And that's when he told me, something's going to open up in California. There wasn't a job open yet, but that's how I got into Ball, was basically through chance meeting with the head of HR. So go out there, meet people, be genuine with them. I'm going to switch gears quickly now. And this is just to talk about plant breeding in general at Ball. So we have 4 business units within the organization, breeding everything from vegetative annuals, vegetative perennials. We've got cut flowers in there, woodies and shrubs, and then also seed propagated plants, both annuals, perennials, and cut flowers. And it's truly a global business with our breeding locations based in Elburn, Illinois. I am based in Elburn, pretty close to our headquarters in West Chicago. We've got a breeding station in Thailand, it's our newest station. One in Santa Paula, California. Another one in California, a couple hours up the road in Guadalupe. This is where I was based before moving to Illinois. There's a good reason that almost all breeding companies have some footprint in that part of the world. It's because it's beautiful weather that you can create high quality seed almost all year round. then you can't think about being in a ornamental business without having exposure to the Netherlands. Then in Costa Rica, that's one of our biggest production farm, but the farm on the photo here is where we do breeding. So we have a breeding site there. I also oversee breeding here. You actually see in this little photo is an impatiens trial. And then West Grove, Pennsylvania, so with star roses. That's the breeding locations within Ball, and this will be a freebie if anybody is interested in breeding and want to apply for a job. If you ever get to the interview, I will ask you who are your customers. So I'm curious, who do you think are your customers? Let's say you're a breeder for a seed annual or an ornamental. Anybody wants to venture a guess? Don't leave me hanging. Growers? Okay.
Landscape.
Okay, the end user. I like that. Yes, sir. Yes, sir. So there's truly your internal customers and your external customers. So you've got your own manager. Don't ever forget that you have to sell yourself to your boss and make sure that he understands that what you're doing is the good stuff. You got product managers, again, still internal in production. Say if we're doing seed, if you can't make seed on this product or produce it in a way that still results in margin, doesn't matter how good a product it is, you're probably never going to get it on the market. And then we spoke about the external customers, the growers and the end consumers like your landscaper, your homeowner. And what I tried to show on this little image here is that what you're looking for is the overlap between all of these different customers, satisfy them. And that's part of the reason why you can make a couple hundred hybrids a year and only introduce one, maybe two out of those. We can't always satisfy all of them. So there's parts here on the sides where you still get products, but really the sweet spot is trying to satisfy all of the customers. So keep that in mind. And again, talked about the people part earlier. And as a breeder and doing breeding, you have to be able to collaborate with many different people throughout the organization. And being in ornamentals, I think in bigger ag, this wouldn't be the case. But in ornamentals, you actually have some input across the whole product life cycle. And I really see breeders and breeding as being generalists who have specialized knowledge. And by saying that, you have to be a generalist in your breeding. And I realize the breeding part, this is just a small bubble, but this is one whole job in itself. You are going to interact with technicians, so the people working in your greenhouse, your growers, could be folks in tissue culture. Do they introduce things in culture for parents? Do you work with embryo rescue? Is there a pathology aspect? Are there other basic research questions that's happening? So you need to be able to speak the language of everybody there. And then as you move along through the process, so it's all internal here, and this is kind of more external customer facing. So now you're starting to move out and you start adding trial coordinators and product managers. So big aspect again, being able to speak to everybody and meet them where they are. And that's important for a breeder to be able to do that. And as you move along the change, things change. So now we're adding people like culture research, figuring out if there's any specific requirements on what needs your product have. Is there IP protection involved? Seed technology, is there anything specific to that? So you might work with seed biologists or engineers. And so as you go along, the kind of people you interact with change. Now the grower is highlighted here. So these are your end customers, so your landscape, so the guys who are growing the plants that end up in the landscape or at your garden centers. And then ultimately, you even still get the chance to interact with sales and marketing and product reps. They love talking to breeding because they feel like they get the real, the good stuff, the good information from the breeders. You know the product intimately, so it's important to still be part of that team. I throw this up here is because this is related to the work hard part that I spoke about earlier. I very clearly remember when I joined the company, I had 2 months. We were at temporary housing and was set up about 200 meters from the beach, and life was just good. I remember calling my dad, like, "This is good. I've made it." And his response was just, "You just wait. They'll get it out of you. Don't worry." And it's true. I mean, enjoy what you do. You're going to, especially as a breeder, we've got Dr. Byrne and Dr. Crosby here. Can they attest to it? You spend a lot of time on the road. I don't know a single breeder who doesn't spend a lot of time on the road, traveling to trials and sites all over. So depending the crops you're working, you could be gone from home up to 30% of the year. So that's not necessarily always easy, but it does come with rewards. I've had the chance as a breeder with the company, travel to over 10 countries for work. And that gives you a chance to see things that you don't normally would. So you might be tired and jet lagged and all of these things, but take half a day and go see some of the sites. This was out in Thailand. Take some time on the weekend and try fishing in a country you've never fished before. This, for example, is over in Italy. Went to do grower visits over there, and it was about an hour and a half drive back to the hotel. And we've already been on the road for 6 days at that point. Slept in 5 different beds. By then, you're tired, but you're like, all right, guys, why don't we just take the long road back? It's going to take us 3 hours instead of an hour and a half, but you get to see the Dolomites, and we end up having a really nice dinner. So take advantage when you can. Make the best of it. Spend some time in Venice or go to France. Visit growers, see these things, but take a chance and go see a castle, see a nice site on your way to the airport. But the important part is the people that you're traveling with, your colleagues, the people you meet. This is going to be another one that I'm going to throw in there for anybody who ever wants to apply for a job. One of the things we discuss afterwards when candidates come through is this somebody you'd feel comfortable traveling with? Like, is this somebody that you would be okay spending a couple of days in a car with and having dinner a couple of nights? So again, what I said before, don't be a jerk. It really goes through. So the people part is the important part. You get to see some fun stuff, but the really thing is meeting people and working with people that you like and respect. So I'm going to switch gears to my own journey a little bit. Starting in industry was a big adjustment for me. I don't know if other people experience the same thing, but I had my research projects. I thought I was busy. At least I like to tell myself that in graduate school. But for example, I started on the 10th of February. There was a nursery flowering already from the previous breeder who was about to leave. 1,500 lines were flowering. I had to start evaluating them, not knowing what I'm doing. Start taking notes, not screw it up. My boss told me, you also have to be in the Netherlands at the end of February for a very important trial. Figure out how to get a visa, because the South African passport at that point, traveling on that is not easy. And so it was just, boom, you just jump in. And that was just one crop. You start off with 4 different species you're working on. As soon as you feel comfortable that you know what's going on and you start relaxing, something change. Either your programs change, your goals for breeding change. Hopefully not too often, but the market change. Somebody retires and you pick up another program, or you shed something to something else. So it's never static. So if you're looking for the kind of role where you want to have high stability in what you do on a daily basis, breeding at an ornamental company may not be the right choice. The fun thing is, though, that it does result in a continuous learning. So I still haven't changed as far as, I don't think I can ever drink everything. So I think I'm same with that Labby there. And so we spoke about this a little bit earlier, my own journey. And so I started as an associate breeder, became a full plant breeder, then senior breeder, and now regional breeding director. And part of, and I don't think I would be able to verbalize that earlier, but part of what you do is what kind of problems are you solving? So initially, you solve problems for yourself in your own programs. Then you start solving problems for maybe your team, your facility, your whole department, and then maybe for the whole company. So that's basically what happens. As you move up, you start solving larger scale problems. So I think if you don't take as long as I did to learn that, that will help you in your career, is to look at what kind of problems are you solving and who are you solving it for. So try to focus outside of yourself when you can. So yeah, so as a regional breeding director, I don't get to breed many crops anymore at all. I only have one left. Just getting back to the people part, when I started out in California, I was by far the newest member of the team. And what that meant is almost every time we had an intern, they were just assigned to the new guy. So I was, my wife said, I was the intern whisperer for a couple of years. But the fun thing is you get to meet students, you get to learn what they're working on and kind of see how they progress in their career. This is just Mary Lewis highlighting one of the interns we had. She's now a successful breeder with Syngenta flowers. What happened was that after interning with us, she asked me to serve on our graduate committee as she was going through grad school at the UGA, which was a great opportunity. I may have gotten more out of it than she did. I just didn't tell them at the time because it was so fun to stay involved with the students and learn from it. And the added benefit was, never expected it, but I got my name, on a couple of papers with work that she was doing. So that was a real treat. So again, just want to highlight, it's the people part. So this was at Cultivate 2 years ago, and Mary was nominated for the GPN 40 under 40. So some of the things that I've worked on. Who can name all the crops up here? First one, Viola, all right? Oh, come on, y'all. Vinca, yep. This one is a little more tricky and it's a little less known. So that's Cuphea. These are actually 3 different types of Cupheas. That's come on. What is the blue pigment that everybody knows? Delphinium, right? Delphinium. That's where the Delphinium comes from, and then that's a phlox, verbena, Zania, and a pentas. But the reason I threw them up here, these are all crops that I had my hand on at some point, some for a long time and some for just a very short period of time. But all of them, looking back, had an important part of what I learned. So I'll just jump in right here with the violas. The main market for them is Northern Europe. At that point, it was Northern Europe, UK, Japan, and China were it's still the top 4. It's just the order of which countries' importance have changed a little bit. But that immediately allowed me to learn the international seed market or ornamental plant market, which is not necessarily common. So immediately I started learning the international market. I'll skip Vinca for now. This little Cuphea, we probably made about tens and tens of dollars in sales on this one. But I could turn, this was a little 2-bench project. I could turn 3, almost 4 cycles a year breeding this thing. And so what it allowed me to do is get an introduction really, really fast. Normally it takes quite some time. But if you can turn 3 or 4 breeding cycles a year, you can actually make progress. And this is a brand new species for us from seed. So you learn, how do you get something new to the market? What kind of seed information do you need to generate? Does it store well? Does it need light to germinate? How does it germinate in the dark? What happens to the fats in the oils in the seed when you store it cold? So all of these things that you don't need to ask for an established crop like Viola or Vinca, Cataranthus, we know that. You don't have to ask those questions because we already know that. All of those things are some things that you need to learn for a brand new species. So bonus. Here again, that's another type of Cuphea. The whole program was bred in Costa Rica while I was based in California. So while maybe at first I was annoyed at the fact that I needed to run a program remotely and travel out there regularly, what it did teach me was how to run programs remotely, understand our production farms, understand how to work in Costa Rica on the species. Um, so when it became time for changing leadership with the Costa Rica team, I had an unfair advantage over everybody else because I was already working with them for 7 years and already knew the system there. So again, it had nothing to do with me. I just got this crop, but what allowed me to then continue on working with that team was getting the job done, working well with the people. So that's what I meant earlier with those icons. This little Cuphea, that was just a lucky fortunate one. Interspecific hybrids, it's Cuphea honeybells. I was making crosses in 2014 in the greenhouse, and my manager came to me at some point and he said, well, you're getting paid a little too much to be spending this much time making pollinations, which now I understand that. But I found one flower out of all these pollinations I made in this interspecifics with one little seed in it, which germinated and became an introduction. I tried to recreate this many times. I've probably tried over 2,000 pollinations since between myself and technicians to recreate this hybrid. never got to do that again. So sometimes you just get lucky. This Delphinium worked it very shortly, but it allowed me to learn a little bit about the cut flower business. Lacranthus was just an old friend coming back from grad school. But here in phlox, perennials, that was working on a perennial interspecific type. Again, so you keep learning something. Disease resistance here, this is how I learned how to think about breeding with the asters. And then Pentas here has a distylus flower system. So how do you make inbreds and how do you think about making inbreds and hybrid products when you're working in a distylus system? So all of these things add to your journey. I left Vinca out because I want to leave it for last. This was a crop where right place, right time. The breeder was very successful, retired. We happened to be almost next to each other. Our office is one office between us, and I had some experience in his products just from walking his greenhouse and his nurseries with him. So I took over this project, and through nothing I did with it, but I was part of the team that brought brand new colors to the market in a class that hasn't seen any color innovation in probably almost 100 years. So that was incredible. This allowed me to start working on mapping projects with developing molecular markers for traits like some disease resistance traits and male sterility traits. So every one of these crops allowed me to learn something different. And moving to the last one, doing a time check there. If anybody ever calls you a pansy, just know that they mean you're pretty tough. These are Violas. They're similar to pansies, just in the Netherlands after a snow. And as soon as the snow starts melting, you see the flowers pop right back out. So this is part of why I really love this crop, is they're very weather durable. So quick introduction. This is a little bit of the science part. And after talking to Oscar and Dr. Koiwa earlier today, I realized that one of the downsides of working on multiple crops, the main goal is to get products on the market. You don't necessarily get the chance to ask deeper questions, which in this case, I think it really would have helped if I slowed down earlier and asked more thoughtful questions. But nonetheless, so it's a cool season biannual. We have 39 different hybrids on the market. So the color range is incredible. And you can make many more colors. They just won't allow me because they want to keep the number of hybrids we sell more limited. Can be grown incredibly cold. It's very cold durable. If you grow them in, say, northern latitude, you can grow them just above freezing. So they can handle quite a bit of cold. It's an F1 product, so meaning similar to how you would think if you're doing any other hybrid product, you'll make inbreds. You make hybrids. Inbreds are maintained from seed. You make hybrids. You have to emasculate, pollinate. So that very classical hybrid system you're using. It's sold as Viola cornuta, but it truly is a multi-species complex with at least these many species involved in contributing traits over the years. When I started out, literature I read was that it was a tetraploid. That at some point changed on me when I wasn't reading literature. So it's now considered a diploid, but previously it was considered a tetraploid. I have a bad habit of just treating everything like they're diploids. And through sheer brute force and numbers, just get to where you want to go. I understand it's not necessarily correct, but I do it in any case. So what is the breeding goals? We're breeding for an idiotype. So you don't want to go too early. You don't want to go too late. You want to be in that Goldilocks zone. So I said earlier, we have 39 different colors on the market. And the main goal is to really tighten up that range. So when a grower grows, especially big growers will, I've seen growers say in the Netherlands, for example, that one greenhouse bay is 500 meters long, and there'll be a couple million seed put out in these bays. You want every color to behave the same. They do not like it if they have to wait for the yellow or the red or the orange for that matter. Everything, the more uniform the colors are within the series, the better. And so that's the main goal. There's a lot of competition in the market for violas. Not really a big North American product, which is part of why I chose this, also because I like the crop, but maybe a little bit of education on the crop for people here. In the US, we like big flowered pansies. But the European market actually doesn't want a bigger flower. You want to make sure it stays the size it is. There's a lot of competition, so your big sellers would be, there's a classic 80-20 distribution there where 20% of the hybrids accounts for 80% of the sales, so there's a very long tail. And the big ones, and especially the ones with the most competition, would be white, yellow, blues, orange, and red. I'm highlighting orange here because we're going to take a direction into the orange violas for a minute. And of course, I mentioned that, but you have to have some fun colors. So I'm messing around with tiger stripes with a true black flower. You have to do some things that maybe not asked for, just to keep your own curiosity satisfied. Violas, before 1991, Viola and Pansy that was orange, this is what they looked like. And then Benari came in 1991 and introduced this Patparaja orange. I have no idea, and I have never met anybody or able to find literature on where this dark orange came from. There's no species I could find that brought this in. So whatever they did, how they got to this orange, that's been kept secret. But what the problem was, now all of a sudden this orange is not okay anymore. Now this is what the market wants. And so all of a sudden everybody's chasing this Benari color. And I'll dive into that a little further now. So I took a photo of some of the variation within my breeding nursery. So that Patparaja, the deep orange I showed you, came in pansy. So I mentioned earlier, pansies are not exactly the same as violas. Pansies are octoploids. I didn't say that earlier, but they're octoploids. Violas are now considered diploids, but they will intercross and have fertile offspring. But this is what your color range looks like. So you can go all the way from yellows into really deep orange, and you can even express the color orange in different parts of the flower. And that's part of why violas are so fun, is because you can control colors on all three levels of the flower. And you can, I see it as like a canvas that you can paint with. You can do all kinds of things with it. But when I looked at breeding orange violas, this photo here, this is what I really want an inbred to look like. Very well branched, very nice flower presentation. I don't ever seem to get there with orange. They still have these elongated stems. Flowers tend to be on the large side, poor branching. It's just when after a couple of years of looking at them, I just felt like, oh, they kind of look like small pansies to me. Um, so moving on, the reason why this would be important, orange is a top selling color. Right now, it's probably our 5 or 6 selling color. But if you could move that orange flower onto the habit of this other little inbred I showed here, you could probably double the sales or more in orange for your company, which would mean you would take the whole market of orange pretty much until somebody else rips off your genetics, but at least it gives you a couple years head start. So that's why it would be important. And again, this is where it would be nice if that's the only thing you're working on, but it's not. So you have about 5 or 6 breeding goals just in this crop, and then you have maybe 4 other crops you're working on. So going deep is not always possible. But what we did was, okay, how is orange made? We found out that if you looked at these 2 oranges, and we did others, I just grabbed some data out. Orange was the only color that accumulates lycopene. So over here, and we saw that orange is also associated with high lutein accumulation. So at first I thought, okay, maybe what we do is to create variation, I'm going to cross with things that accumulates a lot of lutein. So like this primrose, for example, has a nice lutein accumulation. Let's cross with this to see if the orange we recover out of it is darker. So that was part of the approach. Over time, last couple of years, I've made over 281 different kind of crosses with orange. I believe firmly in maximizing recombination. There are some breeders who would say if you're working with yellow, you cross within yellow to improve in yellow. You stay within your color classes. Certainly makes your life easy. Especially when I started my career, I figured, well, I have plenty of time. I'm going all out. I'm crossing it with everything. I want to break up these linkage groups and see what we can recover. 4-way crosses, I tried a lot because I thought that was the way I can really maximize recombination. You guys understand 4-way crosses? Yep. Crossing 2 F1s to got that. So those, I ended up crossing it to yellow, white, blue, red, orange, and many different ways. Made over 108 hybrids for orange. Not a single one going commercial. The main reason for that is I've been failing in production, either through dormancy or seed yield type levels. So we talked about those customers earlier. Well, my internal customers you couldn't satisfy. But the main problem was that unusual segregation ratios. I get orange back more often when you cross it through yellow. But even then, it's about 3 out of 512 is what you pull out. And then when you pull out the orange, it's not a good orange. This is just a photo from a little F2 population that I took a photo of this last autumn. This is that one F2 here. This is the single orange, and then there was one other orange popped out of this. But so I basically found 2 orange violas in this whole F2. And so again, I'm like, what's going on? If I had the tools, and that's my only thing, I probably would have read more earlier. But what happened was, when you work in this color palette, they behave like violas. You can make progress. You can improve habit. You can work on flower time. There's a lot of things that you can improve. Whenever you get into this color palette, especially this Patparaja deep orange type, it's 1 step forward, 1 step back. To get this color back, you have to back cross to it. You back cross, and you drag back all of those other traits. Poor branching, poor flower form, everything associated with that, you bring right back. So in 2018, a paper was published between crosses between pansy, so Viola expitriciana and Viola cornuta, which I didn't read until a couple of years after. But what it showed was something that I suspected, but I never really properly followed up on. And if you remember, I said that first really deep orange, that Patbaraja, was a pansy, was your Exotrichiana types. The aneuploidy is really common, especially when you're working in there. So if I had the tools that you guys have developed to figure out which chromosomes, and that really brought home to me, I was like, oh, there's just a bunch of chromosomes in there that doesn't want to party. And I haven't fully been able to overcome that. But what I then did was using flow cytometry, just quickly looked at genome size and saw that those orange violas that behave like small pansies, they behave like pansies because they were pansies. They had the same genome size that is typical within pansies. And what we actually did over time, even when you cross to viola, but every time you recover that dark orange, is you recover it on that pansy genome size pretty much. We just selected for really small flowered pansies over time. But I also found, or we found that there's segregation for genome size within some of these populations. So what I'm doing now just takes too much effort to count chromosomes on a lot of things. We're looking to see, based on genome size, if we can cross and recover orange on that viola DNA content level. And that's the plan going forward. I'm hoping to give some results of that in the future. And my hypothesis is that if I can breed on the viola level, that maybe there's a chance to get some recombination. I don't know if it will happen. It might not. It hasn't so far, but we'll see. And this brings me right back to the beginning. I mentioned all these things: stay curious, luck, work hard, people. What I didn't mention earlier is have your anchor, understand what's your anchor, especially when you're working. For me, this is my family is my anchor. This is why I wake up in the mornings to go do my job, why I fly, why I do things. This will be different for everybody, but what I would leave you with is understand what is your anchor, and make sure you are clear on that. And that's what this is what I want to leave you with. Thank you very much for your time and bringing me out here. It was a great treat. You're in the horticulture department. There's plenty of good opportunities to have a career there. It's not just mowing lawns and watering plants in a greenhouse. You can have tremendous career opportunities within the industry. So keep at it and keep the industry going. So, thank you. Yes, sir.
What color was that?
If you look at the tattoo Vincas, it's called tattoo because they have these black, get there quick. They have those really black patterns. And I was exaggerating a little bit. A couple of years before we introduced this, about a decade before that, blackberry came out, which was like a really dark pattern. Before that, it was basically white and lilac were your colors. And then in the 90s, we got red and apricot and a few other colors. But this black dark patterns that went all over the petals in different ways was the color breakdown or color breakthrough. Sir.
As a commercial breeder, how often were you expected to put out new variety for new hybrids?
Ooh, as often as you can. It really depends a little bit on your programs. So if you're starting with the company and you're walking into a project with a full commercial pipeline, so something like Viola or Vinca, it's expected that you keep the program going. There should be products in all stages of the life cycle. So you should have early look hybrids all the way to things that's almost close to commercial. So it's expected that you keep that going. But if you're doing something brand new, people understand it takes some time to do that. So that pressure is a little different when it comes to working on completely new and different species. And I'm not saying pressure. I mean, the main challenge is the logistics. When you first walk in, if you can not screw up sowing dates and trial dates and get seed at the production farm at the right time and show up at your trials, and like if you do those things right, it buys you time to learn the crop. So keep your logistics organized.
Ockert, I have a question about how far Ball is into the arena of direct genetic modification with Casper or CRISPR or whatever, the gene editing approach. Are you guys doing any of that yet?
We have some research scientists within the Ball Helix group, and some of those techniques are definitely being explored. I don't think I can speak to any products that's on the horizon to come out. There's obviously some sensitivity around that, especially as a company that we have a global market, so it's not just North American focused, and so we have to make sure that we pay attention to potential impacts, not just in North America, but also in other parts of the world.
Yeah.
A question about your orange pansy. Do you think it's in a chromosome that doesn't pair or recombine with other chromosomes in the complement? That way you are getting recombination and eradicating that particular trait from other traits that you're not interested in?
I think so. I suspect there's one or more chromosome that there's just no bivalence forming, and
one of the things you can do is to try to induce exchanges with radiation. That is some work that we also did in the past. So you can actually apply some levels of radiation to induce chromosome exchanges. And by doing that, then you could segregate that color trait from all of the traits in the area.
And would you do that in hybrids, or where would you, like, if you were in my position, where would you look to apply that?
Whatever material that has the two traits that you don't want, so lots of seed of that, and irradiate that, and then look for segregants thereafter that don't have that combination of traits.
Yeah. Excellent, thank you. Never thought about that.
So we did quite a lot of work on radiation hybrids material. We often find to get those.
Do you?
No, these are diploids, or they're really polyploids?
Well,
because if they're polyploids, they'll tolerate radiation more than a diploid, right?
Latest literature says that these are diploids. However, we have polyploids with these color traits as well.
And I would definitely work with the polyploids first.
Yeah. Thank you.
With your Vinca work, you had your color breakthrough. How'd you do that? How do those color breakthroughs come if you don't have it to begin with, or where do you get it to make it work?
So some of those things you just have to. On this color breakthrough, I actually don't know exactly what the secret sauce was. I actually don't know. What happened was blackberry was introduced. Maybe I can't remember exactly when, but quite a while back. That came out of Japan, and I do not know how they got that color break. But what I can say is that the blackberry color breakthrough is related to some of these color breakthroughs. And so through crossing and exploring that novel color mutation, we're able to see it expressed in different backgrounds. And that's where some of these colors came from. But where the original blackberry that came out of Japan came from, no idea. Yes, ma'am.
Luck several times during your presentation, and you gave us some examples of the good luck you had throughout your career. Did you ever have bad luck? And if so, how did you deal with it?
Of course. So I'll start with the first bad luck that was quite impactful in my life was I wasn't able to secure funding to go to graduate school in South Africa through various reasons. quite politically at that point. It was just difficult to secure funding, and so that was a big hit for me. But what happened, I started looking elsewhere, and was able to then go to Cornell through that. So I wasn't able to get any funding from graduate school in South Africa, but there was opportunity that. Looking back, it was easy, but at the time, that was a big hit for me. I've had several smaller things like not confirming the seed source before sending a hybrid trial out on certain hybrids, and you show up in a trial. I just assumed my team knew exactly what I wanted. I didn't communicate clearly. I show up at the hybrid trial in Europe with pie on my face because the wrong hybrids are there. So call it bad luck or poor execution of my job, I suppose. But I only did that once. What else? I don't know if it's bad luck or not, because when I look at it, where I'm now, I'm happy with how it turned out. But at some point, I put my name in the ring for a regional breeding director job. It was before I was ready, but I put my name in the hat in any case, thinking I stood a good chance, and that kind of knocked me when I was looked over, and they brought somebody from outside the industry, which annoyed me even more. I was like, what is this guy doing? He doesn't even know ornamentals. I've already spent many years in ornamentals, and they brought an outside person in, and what turned out, he became my boss. That was something a pill to swallow, and then turned out this is somebody that I learned so much from that if he didn't, I don't think I'd be able to do what I do now and the way I do it if I didn't have him as a manager. So was it bad? Like at the time, I don't necessarily know. At the time, it felt like it.
I guess the point, fine, yep, for the young people here, don't get disappointed. Things seem to be bad. It might turn out to be for the best.
Yeah. No, I think you just have to. Sometimes you just have to stick it out. I mean, I'm not gonna lie. There was times where I thought, well, maybe I should get back into academia. I haven't been out that long, and I'd maybe like to get back in it. And I missed the research, and I didn't see growth within my role. But that's the problem if you're focused too much on yourself. You can't see far enough if you're on yourself the whole time. Fortunately, I didn't get the jobs I applied for in academia, and other opportunities opened up.
I think the events of the most recent 101 days would convince you that you made the right decision.
So far, I think.
We have time for questions.
Okay.
In token of appreciation, I'd like to give you a little gift from all of us here at the Hort Department.
Thank you.
Thank you so much for joining us and for giving us an amazing presentation today. And, yeah, thanks everybody to come and wrap it up.
Thank you. Feel free to contact me with questions and things after.
Thank you. Good work. I was surprised to see that you work with, hey,